Hi, everyone. Welcome to today's webinar with presenter Corinne Ramos, PhD, MBA, Director of R&D for Aliri Bioanalysis. Corinne joined the Aliri team in 2020 to focus on developing clinical programs to support innovative research. With a strong background in innovative research and clinical development, she has helped many biotech and pharma companies across the globe increase success rates within their drug development program. Without further ado, Corinne, we are ready for you to start.
Thank you very much, Emily. So I don't have to reintroduce myself. Today, I'm going to go over the visualization of immune cell abundance in tumor and microenvironment to determine the therapeutic efficacy. But first, a little bit of a rationale about Aliri. Who are we? So Aliri, it's a next generation contract research organization company. We have more than 200 employees across 3 facilities. There is one in Salt Lake City, another one in Colorado Springs, and another one in Lee. We combine robust GLP bioanalysis capability to measure the target engagement, PK, PD from tissue and blood, mostly in the US capability. And in Lille, we are doing special bioanalysis that allow the evaluation of multi-omics directly on tissue and at the single cell level to measure the true efficacy of your molecule from preclinical to late stage clinical. Seeing the true drug efficacy is very important, and looking at the importance of spatial analysis in evaluating the true efficacy of the drug, it's becoming very important. By understanding the biological context in which cell and molecule exist, greatly influence their behavior and response to treatment. And understanding this spatial context help in developing more effective and targeted therapy, particularly in complex disease like cancer. In order to evaluate the molecular intricacy at the level of the tissue, at Aliri France, we have a different technological platform. One of them is the spatial transcriptomics platform suite that goes from evaluating gene expression from mid-size to high capability. In terms of mid complexity, we have capability like RNA-scope technology that is basically a PCR in tissue that allows to evaluate 4 different transcripts at the same time, at the level of the tissue, at the level of the single cells, and also, like within the histological region of interest. For more multiplex capability in terms of gene expression evaluation, we have the digital spatial profiler, the GeoMX from NanoString, as well as the spatial gene expression, which is the capability from the 10X, it's called the Vision, that allow to evaluate the whole transcriptome directly on the tissue. And finally, in order to increase our capability to look at very specific tissue region, we also combine the laser capture microdissection capability, which allow to dissect a very specific tissue region and perform the evaluation of the gene expression with that particular region by using NGS, for example. For the spatial proteomic assays, and this is the one I'm going to focus my talk today, we also have different capability from low plexing to high plexing. But before I get into this detail, we at Aliri have an histological lab and we evaluate tissue at the level, like of pathological evaluation, as well as the different assessment of tissue staining. For the low plex, we are using immunohistochemistry technique that can plex up to 2 markers. Our immunofluorescence capability allows us to go to 6 markers at the same time. The imaging mass cytometry, which I'm going to develop a little bit later, allows us to perform 40 markers directly on the same tissue section at the same time. Those technique at single cell capability. And finally, we also have a very high throughput protein capability with the digital spatial profiler for NanoString, as well as a vision from 10x Genomics that allow to look at more than 100 protein, not at the single cell level, but at a very specific region of interest or across the whole tissue. But first, I wanted to showcase the value of high multiplexing protein assay. So here you're looking at a meta-analysis that was published in 2019 that was evaluated on about 16 clinical studies to look at the predictivity of different assays to predict the patient response to anti-PD-1 therapy. And you can see right away from this ROC curve that it's showcased in that particular slide that the spatial multiplex capability has the better, much better high predictivity than the rest of the proteomic assay, or not only proteomic assay, but assays that help, like to stratify the patient for response. Spatial simplex, for example, if you look at one protein on the tissue, it's not going to be enough, as well as the gene expression or DNA mutation in bulk. The spatial multiplex proteomic assay has the best accuracy. The application actually of the spatial multiplex proteomic assay, it's very, very good for immunology work due to the intricate and multifaceted nature of immune response targeted by immunotherapies. So the multiplex immune analysis, for example, facilitate the better prediction of treatment response, the identification of resistance mechanism, and the development of personalized treatment strategy. And this by characterizing with a high multiplexing protein assay, the disease molecular environment, the mechanism of action, or the treatment response. So to understand this application, I'm going to showcase two studies today. The first one is to try to understand the resistance mechanism to immuno-oncology therapy. And by understanding that resistance, try to understand what will be the new strategy to improve immuno-oncology therapy outcome. And by doing this, we're going to look at the spatial immune profiling to reveal intrinsic resistance to immunotherapy by conducting baseline profiling of each patient immune microenvironment. The second case study, it's going to be about the treatment response. So we will look at the spatial immune profiling to reveal the change in the spatial immune signature reflective of clinical outcome. And by doing this, we will conduct sequential sample profiling of each patient immune microenvironment during the treatment and correlate change in that spatial immune signature with the clinical outcome. But first, before getting into this case study, I just wanted actually to emphasize some consideration for using the multiplex analysis in immunology. First of all, some technical complexity that's inherent at building this very high multiplex protein signature directly on the tissue. Data analysis is also becoming a huge point in terms of mining all these different marker data in order to showcase, like very specific cell phenotype or immune cell that's going to be relevant, like to the study. And finally, I'll brush around, like the standardization of these assays that in order, like to be very more within the clinical trial, we need to build, like some robust assays that are very reproducible. First, the technology that we're using for high multiplexes single protein assay, it's a standard biotool Hyperion imaging mass cytometry platform that allow to measure target and pathway modulation. We can plex with using that particular technique platform, which is metal-based immunoassay. We can plex up to 40 markers directly on the same tissue section. In contrast with fluorescence, because it's metal-based assay, there is no bleed through from one channel to the other one. So you can really multiplex at the same time 14 marker. It's also a very customizable platform and also very easy to use in the sense that there is no specific protocol actually to use that particular platform. If your antibody works for immunohistochemistry, it will work potentially on the imaging mass cytometry. So first, like you design your assay, and I'm going to showcase, like a validated assay that we have developed, like in the years. And after that, you stain, like your tissue section with all these different metal tag antibodies. There is an ablation that's going to take place. And the metal signal from each antibody, it's going to be reconvoluted onto the image. That's what you see, like at the section 4 in that particular slide. After that, there is a work of image segmentation, and I'm going to get around it, and statistical analysis to perform the identification of all the different cell population within the study's tissue structure. Here, the immune signature that we have developed over the years, and we still, like it's not a fixed, like immune signature, but at this time, like it's something that we have optimized over the year, and that enabled, like the comprehensive analysis of the tumor microenvironment at a subcellular resolution. So far, it's comprised of about, like 34 protein, but as I said, we can go up, like to 14, and we are working, like toward enriching that particular immune signature in order to palliate all the different immuno-oncology molecules. This particular signature so far is composed of structural markers that recognize the different immune cell type. We have, like marker for the B cell, the T cell, the natural killer cells, the macrophage, and the neutrophile. But in addition to it, we also have functional marker. We have, like marker that can do the difference between an M1 macrophage from an N2 macrophage. We have activation marker like the CD69 or the CD25. We have, like marker that also going to be expressed at the same, like cell type population, but allow us to recognize within that cell type population different subpopulation. And in addition to this, since, like this immune signature, it's very often applied to immuno-oncology program, but also, like to immunology program in general. We have, like immune checkpoint inhibitor as well as exhaustion marker and stroma and vasculator marker, just, like to recognize, like the complexity within, like the whole, like microenvironment. As I said, data analysis is very important when you have to mine, like all these different markers onto the same tissue section. And at Allied Reference, we have developed workflow to allow us to characterize and classify all the different, like cell types that are visualized, like by these different markers that we're applying. By using some digital workflow that we built. The workflow starts with cell segmentation, and we use deep learning algorithm to recognize the nuclei based, like on DNA staining to perform what we call a cell segmentation. This cell segmentation, it's really the base of getting, like the best accuracy of classification and phenotyping along the way. So that particular aspect is very important. And we are always, like at Anui, improving the way we are segmenting the cell by incorporating additional marker of membrane, for example, to recognize very specific cell within, like the microenvironment. After that, we can perform a direct definition and thresholding of the positivity of all the different cell type based, like on their marker staining. But beyond, like we can also do some tissue classification and perform automatic classification of all the different regions within the tissue in order to compartmentalize as well, like our analysis. After that, as I said, the threshold of positivity, and we can start, like the work of visualization and phenotyping. As you can see, like everything has been done using either QPath or a Python algorithm that we have developed in-house. And the last point, which is also very interesting, is the way we are verifying all this data. We are building matrix table that represent the frequency of biomarker co-occurrence to really verify the accuracy of our classification. This is what we will call our customized algorithm and our customized workflow. But we also have something, like that it's more exploratory that help, like to understand if there is, like new subpopulation of cells or new markers that are very close to each other that's not going actually to be, like from the cell type, but represent some hub of particular interest. And we base this exploratory workflow on the clustering aspect. And you can see, like the UMAP here that represent the different cluster that you will get, like outside the staining. And after that, we start performing the neighboring analysis that will help us to understand what's the dynamic interaction between all the different cell types and how close they are with each other and how they talk, like with each other. As I say, like the technical complexity of building a very high multiplex signature in terms of proteomics, you always have, like to make sure that there is a specificity aspect that it's really respected and you're really looking, like at the right, like target. In addition, when you are performing metal type-based antibody, the addition to the metal to the primary antibody that's going to recognize your antigen can be also a little bit tricky in terms of keeping that specificity for the target. So for everything that we're analyzing, we always, performing a dual immunohistochemistry or immunofluorescence staining in order to make sure that when we bind, for example, here you have an example of an antibody against CD3 that has been binding, like with a metal, and on the left, an immunohistochemistry classic experiment with a secondary antibody detection. You will respect actually that specificity of localization of your target. And that complexity, as you can imagine, is becoming increasingly increasing, like with the multiplexing aspect. So that's why at Allumy, we are building step by step, like the immune signature that allows, like to keep along the way a total control, like of that specificity and optimization of the signature. Talking about the standardization, we're also looking at the precision and the repeatability aspect of all, like the immune marker, specifically when we want to compare pre and post patient, for example, or patient, like from the different cohort. We want to make sure, that we understand the variability of the marker that we're looking at, and we are completely comfortable actually with that variability or not, depending, like on the marker we're looking at. Here you have an example for the from 3 markers that were, like in the immune signature that I showcased earlier. You have the CD68 for macrophage, the CD20 for the B lymphocyte, and the CD8 for the T lymphocyte. And what has been performed here to look, like at the robustness of our assay was to compare the same region of interest selected in the tissue and equal 3, and those triplicate were then performed on different day and different operator. And you can see by the table showing the percent coefficient of variation below that there is, like some variability, but that variability, like it's not, like as big as you can imagine. And it's really within, like the standard for clinical trial. The concordance as well plot that you have, like on the right for B cell and T cytotoxic cells show that you don't have, that much variability from one experiment to the other one performed, like on different days. And finally, the case study. So the first case study, as I said, we're looking at the intrinsic resistance of immunotherapy. So what we are looking at here at FFPE metastasis prostate baseline biopsies. So those baseline biopsy were part of the clinical trial. The patient were treated with a checkpoint inhibitor. In that case, it was the CTLA-4 as well as the PD-1. And we didn't see any benefit. So by looking like at the profiling with the immune signature, the baseline of those patients, we can start understanding what is the resistance mechanism that didn't allow the effectiveness of this immunotherapy on those particular samples. Here we have performed 30 patients that were staying, like with the immune signature that I showed you before. And we're looking at different, like side of the tumor. We can look at the interface of the tumor as well, like the tumor core. Because we cannot represent the whole, like a 34 marker at the same time. You're looking here at the sample stain, like with the SMA, the collagen type 1, the pan-cytokeratin on the left, and the HLA-ABC on the right. So here the result, and by applying directly, like the exploratory workflow that I showed you before, so that means, like the clustering, you can see that right away the microenvironment is showing the drug resistance. So what is characterized, like by this UMAP, it's just there is, like a lot of CD3. Of a lot of DD3, and also, like depending actually, like on the metastatic location, because it's something I forgot to say, but there are, like multiple locations from brain, lung, a lot of bones as well. You can see that there were, like some tumor that presented some CD8, but not, but it was not, like homogeneous across all the tumor. But in addition to this, there was a very lack of both the Treg presence and also the checkpoint protein expression of anti-CTL4 and anti-PD-1 target therapy. So from that experiment already, we could have assumed from the very beginning that those patients will not respond to anti-CTLA4 and anti-PD-1 targeted therapy. What the specialty, like is bringing actually from that particular experiment, that first of all, you can see, like the tumor microenvironment of all the different metastasis location, and you can really pinpoint, like what type of cell type, what the distribution of it. So in addition to the lack of checkpoint expression, what we have also realized during, like this particular experiment, that the T cell were totally unable with engaging with the tumor antigen. There was a lack actually, lack of HLA-DR on the macrophage, and there was no engagement of the T cells with the tumor antigen, as well that the T cells were in totally tumor excluded topology phenotype. So based, like on this experiment, now we have a more understanding of the mechanism of resistance, like on those particular patients using that special immune signature. But we can start, like to strategize about increasing the antigen penetration or alleviated CD8 excluded topology to increase, like the immuno-oncology, immunotherapy benefit. The next case study that I wanted to showcase today are more about the therapeutic response and the advantages of using very early in your clinical trial this type of immune signature in order to understand if there is like a benefit and there is like a therapeutic response and a benefit for the patient. Here you're looking at some sample from a clinical trial. Those are FFP, renal cell carcinoma metastasis. So we provide the screening, so the baseline before the therapy, and we provide the week 6 biopsy with the immune signature. Those patients were treated with checkpoint inhibitor. So basically, as I said, like we had 2 time points per patient that were staying with the immune signature. And here an example of one of the patient, for example, patient X, we're going to call it. So you have, like on the top, the H&E evaluation, and on the bottom, you have, like the immune signature. It's more, than the 4 markers that are presented here. It's about, like 34 markers that were applied on those particular biopsies. But right away, like looking at the particular patient, we've seen that the screening didn't show that much infiltration of the T cells compared, like to the week 6, that has been treated. And indeed, when you look at the T cell infiltration for those particular patients, so again, like after the digital workflow has been applied to those immune signature, you can see, like everything that has been digitalized, and you can really identify the different phenotype within those biopsy. The sharp pie is showing, that you have an increase with an increase of the T cell population within the 6 week upon the treatment. When you look at the differential cell phenotype upon treatment, you have clearly an increase of the T cells as well as the CD8 T cells. What is interesting in that particular patient that we have seen as well, an increase of the fibroblast. So that patient was characterized by an increase in CD3, which leads, and we know, to a favorable outcome. So basically that patient responded. Expression of checkpoint inhibitor is still pending, so we will, like release the result as soon as we will have it, because we want to understand, like how the checkpoint inhibitor in that case dynamic can be fine, like the signature that we have right now for predictivity of anti-PD-1. And for that particular patient, here we're looking at another patient from the same, like clinical study. So we're looking again, like a metastatic renal cell carcinoma, screening patient on the top, screening tissue on the top, week 6 upon treatment at the bottom. And what we can see here that based, like on just the visual, like for the T cell infiltration, there was not much, like T cells at the screening, but after the treatment, we don't see, like the blast of T cell infiltration like we have seen, like in the patient before. So here the patient characterized by an increase in myoblast, and that did not lead actually into the favorable outcome. So we didn't see, like an increase in the T cell carrying, like the drug target, but we've seen, like and that could be, like of particular interest, an increase, like of myofibroblast after the treatment, which might be, like also interesting to look at in the sense for resistance, like to immunotherapy. So that's being said, what we have showcased today, it's how we can really identify and evaluate insight into the treatment response very rapidly. So we can advance, like drug to larger trial by early assessment of the change in the spatial distribution of immune cell in response to treatment. And that change, we can apply it to phase 1 or phase 2 clinical trial. So in order, like to make very fast decision regarding the progression of the drug candidate to later phase of clinical development. That being said, I wanted actually to thank everyone for joining, and I think the floor is going to be open for questions. So thank you very much. So I think we have a first question. It's what's our experience outside of oncology? So our experience actually, we have, like quite of experience of applying this immune signature to different disease in chronic disease or inflammatory skin disease, for example. So we have, like experience outside the oncology field. So I don't really see any other question coming. So I guess, like we're gonna close that session. So that being said, I thank everyone for joining today. Oh, sorry, there is a new question. I don't really see any other questions. So I guess thank you very much again, for joining today. And I wish you happy holidays and see you next time. Thank you.